proteins are probably one of the most well-known, well-studied macromolecules. It's the macromolecule that most people associate with muscles and other forms of athletics. But what if I told you proteins do much more than that, and quite a bit more, honestly? See, proteins are one of the most diverse functioning groups of all the macromolecules. Their number of functions list everywhere. And today, we're going to discuss that. Today is Big Orange Biology. I am Mr. Jennings. Class is in session. The proteins can do the following. They can act as enzymes, which are basically there to help speed up chemical reactions. They can act as defensive proteins, like antibodies and toxins and venoms. They can be hormonal and regulatory, such as insulin. They can be receptor proteins, which are going to be outside of the cell membrane. They help pick up and respond to cellular signals. They are storage proteins. So you've got some amino acids that you need for later. You're going to put them all in one big storage protein for a later date. We have structural proteins, which provide stability, such as muscles. We have transport proteins, which carry substances like hemoglobin. And we have genetic regulatory proteins that basically like helicase, topoisomerase, DNA polymerase, RNA polymerase, and many, many, many more that regulate when and how and to what extent a gene is ultimately going to be expressed. Proteins are involved in every facet of life. They are one of the most critical, important molecules that we can talk about. So what makes them so unique? What makes them the organic molecule that is different from the rest? And that comes down to their structure. See, proteins have a unique structure compared to the other macromolecules we discussed so far. Basically, a bunch of carbons are linked together. We've talked about lipids and carbohydrates. Proteins do a little different. There is carbon-to-carbon bonding, as seen here with the carboxyl group. However, there's also an amine group here, or amino group. And that's because they're going to create some different structures. They have different kind of linkages. Also, we have this little spicy action here, the side chain. This side chain, if you look for an R on the periodic table, you're not going to find it, not a solo capital R. This side chain, the R represents a functional group. There's 20 different functional groups that can be added there. Each of those functional groups contribute to a unique chemical property of each amino acid. That means there's 20 amino acids that occur extensively in nature that we care about. Now, these 20 are going to be grouped together based on their properties. Now, you're going to go, Mr. Jennings, why do the properties matter? Why do all these little chemical properties matter? They matter because they're going to help determine the protein structure. And if you change the protein structure, you're going to change its function. So we have a whole wide array of functions. And it's all due to the fact that we have a whole wide array of structures. So here are a first part of the amino acids. These are the polar ones. They can be positive and negative, such as arginine, histidine, and lysine. They have these really large polar chains. Look at the nitrogen counts. Look at this. You got a nitrogen here, here. All these little nitrogen and amine groups attached to the end here are going to play a key role in making these side chains very polar. And also they're going to be positive. Meanwhile, you can have a negative polar molecules. We have these tremendous amount of oxygens. And they have a negative charge overall. So that's going to help contribute to a negative polar molecule. Now you can be polar and neutral. So you can have uncharged chains. So we have neutral compounds. We have a lot of carbon-hydrogen bonds. Yeah, we have a nitrogen and an oxygen, but because they're all on the same opposite sides of each other, they kind of balance. Same deal going on here. But all these here are uncharged polar molecules. Meanwhile, you look at the special cases. We'll explain more on these in a moment. Let's talk about the special cases. So glycine, for instance, has a single hydrogen atom attached. And because it's so compact, it can actually bind in tight corners of most protein molecules. Now, proline is the one that was the unique ring structure. So the big benefit of that is if you have something that's really difficult to rotate, you can put it in, if it goes in places where you want them to be stable, they can help stabilize the overall protein in the process. Now, cysteine here has a unique thing because of its sulfur-hydryl groups attached. Because you have sulfur-hydryl, the hydrogens will often just bounce off here, as we see here. When the hydrogens shoot off to form 2H, you're going to have sulfur to sulfur. Well, sulfur really wants to form another covalent bond, and that's what's going to happen. You're going to create a disulfide bridge. And if you have a protein that's in a big chain, so you're going to get a nice little curve going on. Now, amino acids are going to be linked together in a condensation reaction to form what's known as a peptide bond. And this happens in the direction of amino to carboxyl direction, as we see here. So the amino group is going to have the two hydrogen, and the carboxyl group has this extra oxygen. The hydrogen and the oxygen combined to form water, as we discussed in class already. This creates the peptide bond. And it's going to keep happening from nitrogen to carbon the nitrogen to carbon section from amine to carboxyl, and it's going to keep adding on more and more. So we would have another one add here, and it keep adding and adding. So it's critical to understand the direction it travels. This does matter when it comes to creating the protein. So protein sequences are definitely more than just a string of amino acids. They're actually 4 levels of complexity. The basic level is just that. It's the amino acids in a row. Once they're linked together with the peptide bonds, you're going to get a primary structure, as evidenced by here. So you have your different little amino acids. As you see, they're going to be attached. This is your amine group. That's your carboxyl group. And you got a nice little package going on. Now, because of this, though, we have some serious intermolecular forces about to come into play, leading to a secondary structure. So what's going to happen in secondary structure, you're either going to get an alpha helix, this tight coil, or a beta pleated sheet, which is what it sounds like. It's a folded platform that's going on here. That's going to create a unique shape. Now this happens due to the hydrogen bonding that exists within the amino acids themselves. So you have the amine group and another hydrogen, and the carboxyl group with another hydrogen. They're all going to interact with one another, as evidenced right here. So as they're going to keep interacting, they're going to form these hydrogen bonds that's going to cause the protein to contort. So you're going to get an alpha helix or a beta pleated sheet. Now we got to count for all the different little forces that's going to go on here. So this is when you have multiple beta pleated sheets, alpha helixes, etc., starting to attach to one another. Now the interactions between these R groups are going to determine this tertiary structures. Things like disulfide bridges are going to take polypeptides and fold them together. Hydrogen bonding are going to help stabilize those folds, but also is going to keep contorting them. Hydrophobic side chains may try to go off to their own little side. You can even have ionic interactions between the R chains where they have forming salt. You can get salt bridges going on between these proteins because the proteins are so large, water can't get in there to disrupt it, so you're going to get salt forming. This leads to a very complicated looking mess, as we see here. This is known as a quaternary structure. This is when you have 2 or more polypeptides. So basically, all these intermolecular forces are going to cause these multiple polypeptides to link together at once. So tertiary structures like a bunch of alpha helixes and beta pleated sheets kind of folding and contorting due to bridges and such and hydrogen bonding. Well, what happens when we take a bunch of this in the same amino acid, same polypeptide chain, and combine it with a bunch of polypeptide chains, and they're going to fold and twist on themselves by the same forces. Now, secondary and tertiary protein structure does come from the primary structure. And you can alter the primary structure by changing a few things. And the process of doing so is called denaturing. Because you alter the overall shape, you're going to alter the secondary and tertiary structures. Because secondary and tertiary structures are controlled by these different forces. So if you can take out these intermolecular forces, you're going to disrupt the secondary and tertiary. So protein can return to normal sometimes whenever the chemicals or temperature is removed. Because most of the time the protein has all the information necessary, as long as the primary structure is intact. But you can totally mess with its secondary and tertiary, and in the process really affect the quaternary structure by adding one of these factors. Now these factors include temperature, the change in hydrogen ions. This is known as acidity, by the way, if you're not chemistry based yet. So acidity, the high concentration of polar substances, but too much water in there. You can put other polar compounds, and nonpolar substances like oils and other hydrocarbons. They all can have an impact on a protein structure. Now, proteins also interact with other groups. So R chains, because of their own little chemicals as well, they can form weak interactions between groups on the surface of another molecule outside of the protein. So proteins can end up attaching to other complicated things. And because of this, the tertiary structure will change because all these R groups, remember tertiary is determined by the R group, and if the R groups are different, you're going to change the tertiary, and you're obviously going to change the shape. And proteins can be modified by covalent bonding of the chemical group in this side chain. So you can get one or more of amino acids linking to other things by covalent compounds. So you can have hydrogen bonding, covalent bonding, sulfide bridges, all this going on to outside of the regular protein. And all of these have a great impact on the overall protein's shape and thus its function. So in conclusion, proteins are one of the most complicated, most diverse groups of macromolecules that we'll ever talk about. You have 20 different options, and each of them have their own unique interactions because they have their own kind of chemical personalities. It's like having a classroom full of students and you have to balance how they all interact. No classroom's ever the same because of that. So you have all these unique interactions going on. You're going to have formed these own unique bonding and other intermolecular forces. That's going to change the overall layers of structure. And because there's so many different things you can do to that layer structure, so many different amino acids that can be formed, you're going to get all these different diverse functions of one macromolecule. Alright, that's all for today. I appreciate you all watching and listening.